joining us today for the ICM workshop. Today's workshop is about the new features in our very latest version 3.8. My name is Andrew Ori and I'm a senior research scientist here at MolSoft, and if you have any questions after the workshop, my contact details will be shown at the end. If you have any questions during the presentation, you can use the text to the chat in the webinar to ask me questions, and I'll try and address them during the webinar. If not, we have a time at the end to answer the question. Okay, the key topics that we will be covering are the easy 3D stereo viewing using Anaglyph Stereo, a new macro called MolSkin, which produces publication quality surfaces. It's shown here. We just show a quick way of making movies, which is a very easy way to make them from slides. We have incorporated a lot of new search options inside the graphical user interface, including a blast search, so you can directly blast the NCBI sequence database, and you can also search drug bank and pocketome and a few other things as well. We'll also be covering docking and screening. Firstly, we'll look at induced fit docking using the SCARE method. We'll look at fragment screening and a new product called MolScreen, which is incorporated, which is a selection of more than 360 models for a variety of pharmacological and toxicology targets. We also have a number of new Cheminformatics features that we'll show. To get the latest version, you need to go to molsoft.com/downloads, and that will link you to our support site, and you'll be able to download the latest version. Okay, we'll start with Anaglyph Stereo. Apologies if you came to the webinar last week. Some of these is repeated, but we now have Anaglyph Stereo. So basically, all you need now is any kind of monitor and a pair of $10 glasses. You no longer need expensive glasses or monitors or any stereo hardware. So you just need cyan and red glasses. You can view the stereo on your monitor, or you can project it. So you can provide glasses to people in the meetings. You can display in stereo, and it's also supported on the iPad and the Android and your Android devices using iMolView. We've also incorporated into the free ICM-Browser and active ICM, which is free, so you can also use stereo in your PowerPoint presentations and on the web. And you can also print out the image, as I've done here, paste it into PowerPoint or print, and you can view in 3D with the glasses. So if you have glasses at the moment, you can probably see this in stereo, but if not, I'll just show you how to do this in ICM. So if you open the graphical user interface, just here I have a SH3 domain. We can display in stereo simply by going to click on the glasses button here, and if you click and hold, there's some options, and you can just choose Anaglyph. And you can see now we have, it doesn't look very nice without the glasses, but if you have the glasses on, you'll be able to see the stereo very well. And just to show you also in active ICM, so here I have a fully interactive 3D structure in my PowerPoint presentation. And to turn on Anaglyph Stereo, you just need to click and hold on the button here and choose Anaglyph Stereo, and you can see that it's in stereo. I'm glad some people have got glasses on. That's great. So now we are going to move on to the next thing, which is MolSkin, which is a brand new macro which allows you to prepare molecular surfaces. They're generated with assigned colors and occlusion shading by default, so it allows the depth rendering, which gives a better perception of the depth of the pocket, for example. Carbons of each molecule are coloured by its own consistent colour. Labels, ribbons are coloured accordingly. But the key thing is really the contact patches. So it automatically colours the patches where there's contact between two molecules. And also the labels are brought to the surface so you can see them. So go back to ICM. To do this, we go to you load a molecule. So I went to the search tab. I loaded 1N5Z, and to make the surfaces, you right-click, skin mesh, and publication view. It takes a little bit of time to prepare. And so by default, it's made two meshes. It's annotated here in a table which color represents which molecule. And to see the contacts, we'll just undisplay the surface for the peptide of this SH3 domain, and you can see that the regions are colored accordingly. There are also other coloring options. If you right-click on the ligand, for example, go to skin mesh again, you can color you can add your own coloring by proximity to the molecule, or you can make selections in the receptor and color those selections your own preferred color. So just an example here. Okay, so the third, these are sort of graphics new features, and we did cover them last week, but I just want to quickly show you how you can quickly make movies now. We've always had a screen grabbing movie. If you click on the screen, the movie reel at the bottom, basically it records everything you do in the graphics window. But if you want maybe a more controlled way of making a movie, you can make slides, and then those slides will be automatically dumped into a movie by ICM. So if, for example, we like this viewpoint, we got this slide, we make a slide by clicking on the camera button. We have one slide. Maybe we want to see the full structure without the surface. So we make another slide, and then we could just maybe color the ribbon. So I went to display tab, click and hold on the ribbon, you get different coloring options, and just to make another slide really. So now we have 3 slides. Now to dump this into a movie file, we right click on the slideshow button, slideshow option here, and we choose export as movie. We can include smooth transitions between the different slides. We can also include a blend transition, choose a number of still frames between each slide. And you can also include rocking and between each one as well. But just go OK, just go yes, and it's overwriting an old movie file. And now ICM is making that. So now the movie is prepared. You can just view it in standard view on your desktop. So it's a more controlled way of making a movie. Okay, so now we'll move on to the next thing. So the new search tools that are incorporated. If you go to the search tab, you'll see a variety of new buttons in the drop-down option. Now we've made it easier to, if you're looking for maybe a template for modeling, you may want to perform a blast search of the PDB in order to find a sequence similar to make a model. So that's fully incorporated. You can search the NCBI database. We also have a link to the the Pocketome, which is an encyclopedia of conformational ensembles for all druggable binding sites that are in the PDB. This is prepared by Ruben Uppergein's group at UCSD, and Ruben is the founder of MolSoft. We also have a direct search of drug bank. So you can, drug bank combines detailed drug data with comprehensive target information, so it's a good way of drawing out new compounds. And we also have a link here for searching UniProt and downloading UniProt sequences. So in ICM, to do this, just going to delete everything. You go to the Search tab, and we just start with searching the Pocketome. So we click and hold on here, and we choose the option Pocketome. Then you can search in different. You can maybe search by PDB code. I'm just going to use protein name. Just type in a string and search. This brings up a table of search for adrenergic, and you can you can just download the Pocketome hit here, and you see that we have all the structures of the adrenergic receptor that have been deposited in the PDB. But the advantage of the Pocketome is that all of these structures have been superimposed and around the binding site, and so you can analyze the contacts between the ligand and the receptor. If you wanted to do multiple receptor docking, everything is really set up for you here. Everything is, if you wanted to, multiple receptor docking is a way of incorporating induced fit into your docking screening protocols. So that's one way. You may also want to find in drug bank, for example, compounds that bind, chemicals that are known that have been annotated as to bind to the adrenergic receptor. So this is the beta 2 adrenergic. We just click on here with a search string. This searches drug bank, and then we get a chemical spreadsheet with all the chemicals that have been annotated as being adrenergic binders. You can click and go to chemistry and convert to 3D and display them, or you can make your own. If you like particular ones, you can make your own smaller database by going copy selection to new, and just give it a new name, and then you have a new table here. Okay. So the other new option is to search maybe UniProt. So we can, if you have a UniProt ID, you can type that in, CHK2 _ human, for example. So this reads in. So in the ICM workspace on the left-hand side, we have the slides from previously. We have the objects from the Pocketome, and now we have the CHK2 human sequence here. We could read the mouse in, for example. And we can make an alignment by dragging and dropping one of them on the other. So here we have CHK2 mouse aligned with CHK2 human. These are all fully interactive as well, but that's for next week. So the last thing is BLAST. So if we wanted to see if there is a crystal structure of CHK2 mouse, or if there is a decent PDB structure in which we can make a model, we can go to BLAST, type in what you CHK2 human. We know there's a structure, but then you type in the code. You choose what you want us to search, the non-redundant database. But for this example, I'm going to search BLAST search the sequences from PDB. And so I just click on that. This does a BLAST search, which takes a little bit of time. There you go. So I was going to make this is a table, so I was going to make go to Windows and say just try and get the table Windows and then table main, and that will put the table here. Okay, so this is a plot of the query against the sequence in UniProt. This is fully interactive, so if you click here, it's linked to the table with the sequence name and the PDB code for this example. You can also load the alignment for that. So we have obviously a very good hit here, 100%. Okay. So if you double click, it also loads the sequence into your ICM workspace as well. So that's all the search tools, and they're all located in the search tab. There is actually one other one in the PDB search, which is relatively new. You can now search the PDB by substructure of a chemical that's in the PDB that is in complex with the protein. So if you go to PDB search and click on this ring with a pencil, it brings up the editor. So you can draw a query, and we also have this new dictionary as well here. So if you type something, it will automatically find that structure, and you can load it quickly and easily into ICM, into the editor. So it's a drug and chemical dictionary. Just to point out a few other little things in the editor, as you're sketching, we also have a property monitor on the right-hand side. So you can monitor the properties of your ligand that you're drawing, and it's all in as you make an edit, it automatically changes. So sort of color coded by ideal sort of Lipinski rules, I guess, for these molecular weight and hydrogen bond, etc. in donors. We also have a drug lightness score as well. So once you've sketched your query, it'll ask you to save changes, and then you see the smiles are in the search tab here. So you just click on the search, and then we'll get a table with the hits and the query. Here, so we can just, if you double click on, we have here the mol column, the PDB code that this substructure matches. If you just double click, it displays that in 3D. Just need the graphics window come up. So here, found that substructure that we searched, and just double click again, finds it again for a second one. This is for free key UL, so it's fully centered on that molecule that you queried. Okay, so that's the end of the search section. So we'll go back to the PowerPoint presentation. Right, so the next new feature in ICM is fragment screening. So in the distribution of ICM, we now provide a library of fragment-like ligands based on the publicly available ligand receptor complex structures in the PDB. And this structure in this database is available for you to screen. For each fragment, there are multiple examples of complexes that have been solved experimentally. And for each fragment, we have provided scoring statistics and derived scores for native-like or non-native-like poses of that fragment. This has allowed us to make a distribution of energy scores for native-like and non-native-like poses, And then these distributions are used to estimate p-values, predicted bound pose of the fragment. This work has been undertaken by the principal scientist here at MolSoft, Max Totroff. So we screen this. You can screen this database, and you'll get a number of different fragments in the pocket. And we also have a fragment linking option. So once you've identified fragments, you can then search a database of linkers to join those fragments together. So just got a quick example for that. So in this example, we have a kinase structure with a ligand bound. It's one REK. So in order to prepare this structure for fragment-based screening, we need to delete some things. That I've already deleted the water. There's another molecule here that I'm going to delete, which is in the C-terminal. Delete. And so that the ligand doesn't interfere the maps of the binding pocket, we need to move this ligand out of the pocket. So right click on the ligand and say move from object. It appears that it's still there. We now have two different objects, so this is no longer seen in the receptor. It's sometimes useful to leave the ligand there because you can compare the ligand with the fragments that you find, for example, or you can use the ligand to define the pocket, which is sometimes a good idea. Or I'm just going to just show the ligand pocket finder, which will find the cavity, all the cavities in this kinase to dock to. To do that, we need to first, when you read in, you would have gone to one search and type 1 REK. That would have read an X-ray structure. We'd have XR here, and I've already converted it to an ICM object. So what I did was right-click and choose to convert to ICM object. This is already pre-prepared. So the next step would be to find the pocket. Go to Tools, 3D Predict ICM Pocket Finder, and go OK. And we get what we call meshes, which represent the pockets. So you see that we have two in the N terminal, a few smaller ones in the C terminal, but the ATP binding pocket in the N terminal is shown in blue. And to select that one, we just double click on the table. You can see that we made a selection. So with fragment screening, we need to be careful about where we select because we don't really want to find pockets outside of the main pocket that you're screening to, otherwise the fragments can go in there and it just causes a little bit of noise. So it's good to be careful about how you select the pocket. So the next step is to go docking menu. We need to set up the receptor. Click on Set Up Receptor. Choose, give the project a name. And the receptor object is one REK. The binding site is called a graphical selection, and graphical selections are shown in green here and in blue here. And so it's 39 residues. And we'll just go OK. And this, in terms of docking, the first step is to choose where you want the simulation to begin. We have this small probe here, and by default it's in the center of the binding pocket. You may want to shift it, but generally it's best just to leave it in place. Generally, the sampling is stochastic, so it's going to jump around the pocket anyway, but this is where it will start. So go OK, if you're happy with that. And the next step, especially for fragment docking, is to make sure that the box snugly fits around the binding pocket that you've chosen, like that. So then we press go, and you can see it's generating the grid maps for hydrogen bonding, electrostatics, and van der Waals, soft van der Waals as well. So this takes a minute or so to make the maps. So once the maps are made, it will tell you here that it's finished. Everything is placed into, all your docking directories are placed into what's called the working directory, and you can find what is your working directory by going here. Mine's in my C drive here. You can change it before you do docking, and it's placed the files where you want them. And it also means that if you want to if you close your computer, you can always come back to your docking project by going open project, and it will come back to exactly how you left it. So the next step for fragment docking is go to docking, and then there's an option here called scan probes. You can increase the thoroughness, so this is the length of the simulation, number of poses to score for this ligand. We also have statistics, so you can have a cutoff, percentile cutoff. You only display hits that have got good p-values and in a decent percentile range. And then the database, I think if you leave it as default, it will find the correct database, but it's actually in your distribution. So if you're using Linux, it'll be in your ICM home. If you're using Windows, it should be somewhere like in the C drive, program files, MolSoft, ICM Pro, fragments, and the fragment screening file is fragments.icf. Okay, you just go OK, and that sets up the screening, and the screening will start running. I'm just going to stop the screening because I have an example finished. So let's do edit, delete all. So once the screening is finished, you'll get a dialog box displayed saying that the screen is completed, and then a table of results will be displayed. So, and this is your hit list for your screen. So the table has the mole column, an index value, the name of the compound, the virtual ligand screening score, number of atoms, number of flexible bonds, this is the hydrogen bonding energy and the other energies are shown here. But the new columns for fragment-based screening are here. So we have a p-value. So the p-value should take precedence over the score because you want to find a statistically relevant pose. So you can right-click on here and choose sort, and you can sort by p-value. You also have the percentile value here. Receptor confirmation is irrelevant in this case because we're only using one confirmation. So you'd sort by p-value, and you can also view this fragment in other PDBs and see how that pose compares to the one you have here. So to do that, you click on this link, and it will load all these PDBs. So this example has quite a few hits. And also another very useful column is called location. So this column enables you to choose fragments in different parts of the pocket. So you can cluster this column and choose different sites. So I just want to show you. So this button here enables you to toggle that fragment on and off. So here I have one here, and if I go down, this is the other fragment. Just on display. So we have two fragments here. if you wanted, I chose the second one by performing clustering on this location column. So to do that, you select that column by clicking on the column header, single click, and there's a clustering button here on the right hand side, and just go OK. That will produce a tree. You can change the distance by clicking and dragging here on this bar. You can see the changes of the branches. We may want to select a number of different fragments that are scattered around the pocket. So to do that, we would choose a center. So each of these clusters represents a different position in the pocket. So we right click on here and choose select centers by. So right click on the tree, choose by, select the centers by minimum p-value. Just go. There's other things here as well. So if you've got a very large cluster, it will choose more from larger clusters, or you can just choose one from each cluster. So you can play around with those options. Go OK. Then you see that it's chosen a center from each of the branches of the tree, and that's they're highlighted here. So you could maybe copy that selection to another database, another table, for example, by going copy selection to new. Might make it easier to filter out these hits. But the key thing is to look at the p-value here by right-clicking and sorting by p-value. Okay, so then you can maybe compare with the ligand from the crystal structure, and online there's a written example which shows you how to do that display. And then once you're happy with the way it is, the fragments that you have, you would put them into the same object. So you can drag one fragment into the receptor object. Then you go to the ligand editor tab, and you use the option called find linker, and that will search a database of linkers. Sorry, the screen was frozen. I apologize. Just to recap, there's the location here, which is a column which you can cluster. So you can just single click on the location, and you click on clustering here, and there's the other columns to view and undisplay the fragments. There's a column called L columns, which you can click and show it here. Apologize for that. I froze the screen back in. And then in order to select fragments that are in different parts of the pocket, you right-click on the tree and choose select centers by minimum p-value. Yeah. So the table is basically your typical screening hit list table with the energy values and everything, and receptor confirmation, but it has these additional values called p-value, percentile, and example PDB. So if you want to see this fragment in other examples, you click here. And also location, which you can use to find different fragments in the pocket. And then to search the linker database, you go to Ligedit, Advanced, and choose Find Linker, and that will screen a database of linkers for those two. Okay. Next new option is actually it's a method that was published in 2008 by scientists at MolSoft and UCSD in Ruben Abergain's lab, and Max Totroff here at MolSoft. So it's a method called SCARE, and so generally you have a receptor and you have a ligand. You want to dock the ligand to the pocket. Usually there's some steric clashes if this pocket hasn't been solved with this ligand inside it. You have some maybe 1 or 2 side chains that need to move out or move away in order for this ligand to dock well. So the method called SCARE is based on the fact that it scans alanines and refines. So the first step is to make gapped models by dual alanine scanning. So it takes pairs of residues and places alanines there and generates a series of models. And then grid docking is then performed to multiple versions of the binding site. And so you have multiple models here, and you dock to each one using the 4D docking in ICM. The top scoring ligand pose from each grid simulation is then refined with explicit flexible receptor, and the best scoring refined conformation is selected as the final answer. So on a benchmark of 30 cross-docking pairs, the top ranking near native solution was found in 80% of the cases, and this is fully described in this paper here. But it's one of our methods for incorporating induced fit into docking. So in ICM, we can just an example here. So once again, we have an example with a kinase. We have one structure here with a small inhibitor against the hinge region, shown in yellow. This is JNK3. Yeah, and we have the codes here if you want to have a look. This example is also online. We also have another JNK3 kinase here, shown in green. You can see this ligand is bigger, and the crystal structure shows that this methionine needs to move away. This is sort of gatekeeper. Residue moves away a little bit in order for this ligand to bind correctly. So in order to do that, we need to have some kind of induced fit docking. So if we wanted to dock this ligand to this receptor, for example, if we didn't have this complex 1-PMN, more than likely your docking is going to be incorrect because you'd have a clash here between the ligand and that methionine. So to do that, to do the scare docking, we have read in 1-PMV, which is an X-ray crystal structure. We would convert it to an ICM object. So you right click, choose Convert PDB. You would choose Delete Waters. You can optimize hydrogens, optimize the positions of asparagines and glutamines and histidines based on the most optimal hydrogen bond network. Replace the original, we'll just overwrite this X-ray structure. Hide missing side chains. You may want to, depends how you want to work, but if the crystallographer has found has seen 1 or 2 atoms of the backbone, ICM will then build the full atom for that residue. So if you uncheck this, then it will build the full atom version of that residue. But some people prefer not to do that, and you can choose that option here. So that's the way to set it up. Then we would go to, let's have an example already prepared. So the next step would be docking setup receptor, as we did before. We would move the ligand out of the pocket, and as we did with the fragment example, we'd move the ligand out of the pocket, and then we would go docking, set up receptor, build the maps. Or actually, in this case, you can uncheck the make receptor maps, because when you build the 4D, when you do SCARE, it builds a series of models, and then it will generate 4D maps. So the next option would be, after setting up receptor, you would go to flexible receptor, and then there's an option called SCARE setup 4D. So this builds the models with the dual alanine scanning, and it will build the maps for each model. So it takes a little bit of time to generate that. Okay. So once you're happy with that, you would then go docking and then choose what you want to dock. So you would choose the ligand. In our example, we're docking this green ligand from 1PMN. So you would have to take this out of this exostructure, right click, move from object, like that. And then you run the docking, and it will do docking into the 4-dimensional, what we call 4D maps, which is basically a multiple receptor conformation. So you have multiple models with different alanine positions here, which opens a pocket. So once the docking is finished, you will have once again we have this hit list table, which we saw with the fragment. We have fewer columns because we don't have the statistics for this. We have a score, so you'd rank these by score. So right click, choose sort by score. The key column here is, because we're docking to multiple conformations, you'll see that in our SCARE1 receptor we have 15 different conformations of this pocket. So we can view those here. If we expand the stack here, we can click here. And the key residue to keep an eye on is this methionine here, because that's the one we want to move out in order for the ligand to dock. So if we toggle through these, we'll see that we'll eventually find one where here, for example, the 5th one has an alanine in place of that methionine, which opens that pocket up. And same with the 6th conformation. 7th one, the methionine comes back. So most likely, we hope that we find that the top hit would be in maybe receptor conformation 5 or 6. And we do find that the top hit here is actually in receptor conformation 6, this one. So you can toggle that ligand on, and you can compare it, for example, to the crystal structure, which was green. And it's pretty good. This is actually without refinement. So you can refine it to improve the interactivity even more. You go to docking, flexible receptor, and then refine the SCARE solution. And that will have a sidechain refinement in order to improve this pose. But it's pretty decent. It would have been completely wrong if it was in the original crystal structure with that methionine out here. So that's the SCARE method. Just a recap of the other methods for flexible receptor. We have you can choose in the grid maps. You can choose these are other methods other than SCARE. So we have basically four different approaches. We have 4D grids, which is kind of used in SCARE, but if you're in a situation where you have multiple crystal structures, you can dock to those multiple crystal structures and use those multiple receptors as a way of incorporating juice fit. We have explicit groups, so you can choose certain residues you build maps, but you can choose certain residues to be fully explicit, to have full atom rotation and sampling of those of particular residues during docking. So that's explicit group. We also have refinement, which allows the side chains to refine around the ligand. So there's 4 different ways of incorporating juice fit. But the SCARE is a very nice method in order to open up the pocket in certain regions. So it's a good approach. Okay, so we're getting close to the end. The other new product we have is called MolScreen. MolScreen is we have 360 fully high quality, fully validated models, which include models that use fingerprints, 2D QSAR fingerprints, and also models that use 3D atomic property fields, so like a 3D pharmacophore approach. So each method has its advantages. Each model has its advantages. This one, for example, is extremely fast. You can screen thousands of compounds in seconds. It's good for counter screening. 3D approach models are the ligand memory is less, but you have which the speed is about 3 to 4 seconds per compound. And it's good for screening, general purpose screening. So the fingerprint model is built using a kernel regression method, and it's built based on the Chemical version 15, and there's approximately 692 chemicals per model for this. The APF, the 3D models use atomic property fields, and which is a 3D pharmacophore potential implemented on a grid, and I can send you Max Tochrov's paper on this method if you'd like to read it more. So for the APF, there are seven properties are assigned to each ligand. Things like hydrogen bond donors, acceptors, lipophilicity, the size of the group, sp2 hybridization, and then charges, positive and negative charges. And these models are built from superimposed structures from Pocketome, which are taken from the protein data bank. So to run MolScreen, it's very straightforward. You go to, let's got an example here, MolScreen example. So you would go to File, Open, and read in an SGS file with the compounds that you want to screen. You go to the Chemistry menu, and if you have ICM-VLS, you will have automatically included a series of models here. If you want to purchase the other models, you can do, and then you'd have a full model panel here, which is a table of each of the models we have. So we have, so it tells you the name of the model you're screening to, what kind of protein family it's in, the diseases it's involved in. So you select the ones that you want to screen against. For example, like this, you can use Control key and select non-contiguous set, or you can select a range of ones that you might be interested in. Then you go to, then basically you just choose in this table. There's Apply Selected Models, and you run it. It's very good. For the 2D ones, it's extremely quick, and then you'll get a table with the values of the screening. So higher values probably indicate that it's a likely binder to this target. So here we screened against 12, 14 or so different models, and you get a table like this, which you can sort, and you can see the hits are color coded. Yeah. So all these options are in Chemistry menu, and the brief menu model panel here is included in ICM-VLS. So finally, just some new chemistry features that are in this latest version. We now support extended connectivity fingerprints, which are topological fingerprints for molecular characterization. So these are circular fingerprints, and they're usually used in for structure-activity relationship models. We now have a new chemical search dialog, which enables you to, well, we've always had chemical search, but you used to you can sketch your molecule, a single molecule, and then search it against a larger database. But now you can read in a table of more than one molecule and search that table against a larger set and compare the two. Entry keys are now supported. We have a new, we've had drug lightness, but now we have a score called ToxScore, which enables you to predict the toxicity of a compound. So that was built using around 1,000 smart strings, and a demerit score was given to each compound based on their perceived toxicity and frequency of appearance in improved drugs. And the tox score of any compound was calculated by summing up all the matching smart strings. And the tox score greater than 1 indicates that likely that compound is toxic. So this is a useful score that you could run before virtual screening or after. So after, in order to filter out compounds which you might not want to test. So just have a quick example of that. So we just read in go file, open SGF, and then we can insert a column. If you right click on the column header, insert column, function, chemical, and then it's at the bottom here called ToxScore. You just go OK. And you get a column of ToxScores, color coded. Blue is good, not toxic, and you can sort. And you see it coloring by, and it, for those that are predicted to be toxic, there are provided reasons why we think that they might be toxic, according to the model that we use. Okay, so that wraps up the today's webinar. If you have any questions, please feel free to email me or call. Our next webinar is on linking sequence to structure. And let's quickly show an example. So for example, here we have two kinase structures. We have the sequences of them here, and we will learn how to read in sequences next week, how to align them, how to annotate alignments, how to make good quality images of the alignments for publication, and how to link the sequence directly to structure. So we can color, for example, we can color by alignment here. You can see the conserved regions are colored blue, non-conserved are gray. So that will be next week. But if anybody has any questions about today's webinar, please feel free to text and let me know, and I can unmute your microphone and you can ask the question. I'll be here for the next 10 minutes or so if anybody has any questions. But thank you very much for joining us today. Thank you. We had a question about the docking results compared to other programs like Autodoc and another program. Yeah, I mean, there's lots of published comparisons by independent people. There's the most recent competition, I think, was in 2012. I think it was reported at the ACS in Anaheim, and ICM did very well. And the results of that are I can send you a link to that paper. I can try and find that now. But yeah, I mean, if you want a trial version and you want to compare, we can probably arrange that. But the docking we showed today really was fragment docking, obviously, and the scare method for incorporating induced fit. So the comparison of how well that method, the scare method, is described in that paper for a benchmark of proteins. So this paper is the most recent benchmarking results for ICM, and some pretty good. These are results from the competition that I think OpenEye set up, and these are the ICM results for that. Some pretty decent discrimination between binders and non-binders in scoring, and also RMSD for docking and redocking. Just had a question regarding pharmacophore models. Yeah, we have online so that the best way to build pharmacophore models is using atomic property fields. So in the presentation, we